The process of aging is often accompanied by a number of life-altering biological changes that occur within our human body. Naturally, an aging body tends to decline in functioning over time as the body wears out. Previous studies have shown that as we age, our bodies tend to deteriorate both physically and cognitively. According to the National Institute of Health, reports suggest that Canadians aged 60 and older also suffer from insomnia. Our research aims to recover from lack of sleep by targeting the impaired entrainment patterns and circadian rhythms. This leads to a decrease in daytime sleepiness, elevates attention, memory, and mood, and provides an overall higher quality of life. Changes in sleep patterns are commonly evident in their EEGs, with shorter time spent in REM sleep and more time spent awake. Therefore, maintaining good sleeping patterns and ensuring optimal sleep quality is an integral feature of successful aging. NMDA is an ionotropic glutamate receptor. Based on its stimulation, it leads to an increase in the presence or the deficit of glutamate. NMDA subunit NR2B is physically coupled with the inhibition of the RAS-ERK pathway, which plays an important role in light-induced phase shifts of our biological clock. Phosphorylation of ERK via NR2B subunit leads to light-induced resetting of circadian rhythms. We chose to manipulate this pathway because optogenetic stimulation of the RAS-ERK signal will help us foresee which pathways are specifically affecting the suprachiasmatic nucleus. Mainly, this research focuses on elderly mice since they have similar ERK-RAS and SCN pathways, which will be easily translatable to humans. Based on previous findings, we hypothesized that optogenetic stimulation of NR2B can help rejuvenate the impaired entrainment patterns in mice models. We predict that NR2B stimulation will lead to an increase in calcium buildup and activation of the RAS-ERK pathway, which further enhances the resetting of impaired photoentrainment patterns. Keeping the limitations in mind, various external factors such as environment, diet, hormones, and interaction with others will be closely monitored to ensure external validity. We will conduct this research using reasonably priced aged laboratory mice, approximately 18 to 24 months old, which correlate with humans ranging from 56 to 69 years old. Lab mice are ideal for aging studies because of their longevity and small body for easy handling. 80 mice chosen for this study will be separated into 3 conditions: control-control, control transgenic, and experimental transgenic. Efficient groupings of the mice will help ensure the validity of results. Proper research hygiene guidelines will be followed under the Canadian Council of Animal Care. Some of the research methodology includes optogenetic manipulation, transcranial alternating current stimulation, and many others which have been proven to be highly reliable and effective in modulating brain oscillations. To ensure the longevity of aged individuals, various sleep disorders result in undergoing surgeries like obstructive sleep apnea or require long-term healthcare investments. However, our research dwells in simple but effective NR2B stimulation, which can then be mediated into a drug or injection, which is very cost-effective with various other benefits. Additionally, our research will be handled primarily via research assistant volunteers who will help gather data. They will be highly trained in performing optogenetic stimulations and transgenic surgeries and surrogates to increase success rate. This will be both cost-effective and will require less employed staff. If chosen to fund this study, it will be able to maximize its impact on sleep patterns and overall quality of life for all aged individuals, as the study will provide promising results and will further lead to less healthcare costs. One of the difficulties in aging research has been the ability to test experimentally whether changes are strictly gene-specific or physiological. Models used for generating transgenic mice will be unique, as we will be able to probe more directly and identify the underlying biological mechanism that leads to abnormal circadian rhythms. There is a sense of urgency in the field of aging research, where sleep is thought to be a crucial factor affecting other aspects of health. Conducting our research and its future implications will take us one step closer into solving one of the many problems associated with the aging process.